Welcome everyone again to the Green Era Hub webinar series. My name is Barry Crosby and I'm one of the communication officers for the Green Era Hub project. If you've been here before, you'll know that the Green Era Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe programme. And it currently represents 15 EU ERA-NETs, co-funds and self-sustained initiatives in the field of agri-food and biotechnology. We started these breakfast clubs here last year in October, and we've had 4 very successful series so far. The first one was circularity in agriculture. The second series was related to biotechnology to advance sustainable solutions for bioeconomy. The third was sustainable food systems. And the one that we just finished before the Christmas break was precision agriculture technologies for a sustainable future. If you're interested in listening to the talks related to any of the topics from the series just mentioned, our recordings are now available to view on our YouTube channel. Now, for the next 3 months, we're focusing on the topic of plant health and protection, where we will be hosting a series of expert-led discussions aimed at fostering a deeper understanding of plant pathology, integrated pest management, and sustainable agricultural practices. And just a few housekeeping announcements. Please feel free to type your questions in the Q&A chat section, which I've enabled for everyone. I'm going to do my best to address as many questions as possible within the time that we have today, which is 1 hour. If at the end of the session you have a question for either of the speakers, please do remember to address the speaker in the question. Now, today we have 2 panelists joining us, Dr. Sarah Green and Marlene Boterman. We will have our opening session this morning with Dr. Green, who is currently Principal Forest Pathologist at Forest Research UK, where she specialises in tree health and plant pathology. Overseeing the pathology laboratory at the Forest Research Northern Research Station, Dr. Green leads groundbreaking research on addressing tree health challenges in UK forests, particularly focusing on humaceae, fungal and bacterial diseases. In addition to coordinating the UK Strategic Priorities Fund Bacterial Plant Diseases Programme, Dr. Green is also currently involved in innovative research projects, including molecular detection of Phytophthora in diverse environments and investigating natural resistance in tree species to key pathogens. Today, Dr. Green's talk is entitled Early Detection of Phytophthora in EU and third country nurseries and traded plants, which is part of the UFRESCO Phytosanitary Research Initiative. Dr. Green, thank you very much for accepting our invitation for a webinar series. I'm going to stop sharing my screen now, and you can start sharing your presentation. Thank you very much.
Sequence in an aluminum MySeq here, and the individual barcode reads were then put through a bioinformatics pipeline, which we developed as part of an earlier project, which tells us every Phytophthora species that is present in that sample. We're about to publish a paper on the methods that were developed for this project, and that's just about to come out in Plant Pathology. And the pipeline, the Phytophthora pipeline, THABI pipeline, was published and is publicly available as of 2023. So we also carried out baiting of water samples. So the water samples were placed into these plastic tubs, as you can see here, and then bait leaves floated on top. The idea being that any Phytophthora zoospores, because they swim in water, will be attracted to the bait plants and then cause lesions. And then these lesions, if we see them, are then cut out, plated onto selected media. Any Phytophthora-like colonies are then PCR'd and Sanger sequenced and then matched to verifiable GenBank sequences for identification. So those are a quick overview of the methods. I'm now going to run through some results from just 4 of the 6 countries, for brevity. And these were the countries that sent me photographs. And then I'm going to collate all the data at the end to give a sort of an overview. Anyway, in the UK, we sampled 2 ornamental plant nurseries in Scotland. Both of them import plants from overseas. We found 26 Phytophthora species at nursery 1 and 30 Phytophthora species at nursery 2. So that's a pretty high Phytophthora load. We also found Phytophthora species and hosts of concern. For example, we found Phytophthora ramorum, which is a very broad host range, very aggressive invasive Phytophthora, which is killing oaks in the USA and also killing larch in the UK. That was found infecting rhododendron in this photograph here. We found Phytophthora ostracedri, which is another quarantine-regulated Phytophthora. It's killing junipers in the UK, and it's where it's invasive, and it's also killing Chilean cedar in southern Argentina. And so this pathogen we found on these twisted Leylandii, you can see the foliage lesions here, and these had been recently imported into Scotland from the Netherlands. Additionally, we found Phytophoras in plant dumps, including the parent of a hybrid Phytophthora, which is killing alder. And these plant dumps are real hotbeds of Phytophthora. So we found a little bit of an issue there with the plant dumps. We found a Phytophthora new to the UK on Pinus mugo, and also on these two hosts here, we found again invasive Phytophoras that are known to spread in the trade. So moving on to the US, where they sampled two very large, much larger than ours, ornamental plant nurseries. One, they found 20 Phytophthora species at nursery 1 and 7 at nursery 2. In particular, they found the hosts of most concern were blue potato bush, which were host to 6 different Phytophoras, and also rock rose, which harbored 2 quite damaging Phytophthora species. The group in Italy sampled from 2 nurseries, and these were quite different nurseries. Nursery 1 was a small experimental nursery which didn't import plants and had higher levels of biosecurity practice, and they found 11 Phytophoras there. And in nursery 2, which was a large ornamental nursery which imported plants, they found 21 Phytophthora species. And the hosts of most concern, which harbored the highest diversity of pathogens, were Choisya viburnum and ligustrum. And then finally, for this little part of the presentation, results from Greece. They sampled again 2 quite large ornamental plant nurseries that import plants. They found 8 and 16 Phytophthora species at each one, respectively. Their detections included some quite rare warm temperature pathogens. And again, the hosts of most concern were Myrtus, Lavandula, and Azalea, just because they had so many Phytophoras associated with them. So then we did a combined analysis. So we combined the data from the 13 nurseries sampled in 6 countries. In France, they actually sampled 3 nurseries. Everyone else sampled 2. And this included 647 root samples and 364 water samples. So it's all this data from 6 different countries. We identified 65 unique Phytophthora species across the whole project, and 86 samples in total contained unknown or identified Phytophthora species. And then we did some modelling, which was carried out by my colleague at the UK CEH, Louise Barwell. And she did Bayesian modelling to look at the effects of certain factors, and one of them was management practices, and those management practices that went into the models are listed here in red, and I won't run through them, but you can have a look, the sort of thing we were looking at. So these practices were collected for each nursery, and she looked at the effect of selected nursery management practice on the proportion of Phytophthora positive samples present at the nursery. And then she looked at whether Phytophthora communities differed among water and plant samples, and also effect of host genus and latitude. So, cutting to the quick, in terms of effective management practices, basically any nursery which raised a very diverse, the higher the diversity of plant stock, and those which imported more than 50% of the plants at the nursery and used open water sources or recycled water without treating it, which tended to be the case in southern latitude countries, they had more Phytophthora positive samples and much more diverse Phytophthora communities. A quick look at here in terms of prevalent host Phytophthora association. So the host species, the host genera here, which were considered to be of highest concern in terms of hosting all the different Phytophthora species, are listed up on the left here. So you can see Fagus, Ligustrum, and Thuya, and Choisya quite high. And the portion of positive samples, the redder the square is, the more Phytophthora, the more that these hosts contain the Phytophoras listed here. This graph shows the Phytophthora species sensitivity to sample substrate. So basically, the Phytophthora species listed on the left, those that were more associated with root or plant samples are up here in green, and those Phytophthora species which are more associated with water were down here in blue, and those in the middle were equally likely to be found in water as in soil. And this is actually really interesting because it enables us to start to build a picture of the niche, the favored niche of the different Phytophthora pathogens and other species in the nursery environment. And then we looked at the latitude. So again, those species up here, up on the sort of listed here on the left, which are sort of pink, are more likely to be found in the lower latitude, so the warmer temperature phytophthora. And those down here, mostly in the blue, the higher latitude or cooler temperature phytophthora. So again, it's allowing us to build a picture of what we might expect and what phytophthora diversity you might have at different geographical locations. So finally, we were reporting results back to the nursery managers. Obviously, this is very important as the project progressed, and we built in a number of management recommendations which went into our best practice guidance. So number one issue is use of open water supplies is high risk. There's lots of phytophthora found in open water supplies, in particular if they're recycled. If you're going to recycle water or use open water sources, then you need to treat it. Need to control irrigation and improve drainage to prevent runoff, sanitize surfaces between batches, raise more plants off the ground. We've identified high-risk hosts for having lots of phytophthora associated, so they should reconsider trading in those hosts. If they're importing plants, they need to inspect and quarantine those imports away from other stock. We identified a clear need for staff training, particularly around symptom awareness and how to deal with symptomatic plants and get diagnosis. And also, above all, we need really good guidance on safe green waste. Well, how do you actually dispose of your sick and unwanted plants? So these are all built into our guidance here. And final slide is we published this two-sided best practice flyer in 5 different languages, which were then circulated within the different countries that were partners in the project. So with that, I would just like to obviously thank all the partners involved in this. It was a really great project. I think everyone was in agreement. What a good fun project it was, and I would also like to particularly acknowledge my UK participants and partners here from the 3 different institutes. And with that, I would like to thank you for listening.
Interested there towards the end of your presentation about the water management issues, and which seemed kind of like absolutely critical in terms of kind of managing the spread of Phytophthora and kind of mitigating against kind of losses in nurseries and so forth. I was wondering, in kind of following on from that, about the challenges that water management present to nurseries. So presumably for some of the kind of larger nurseries, they'll have the kind of capacity and resources to be able to implement those changes that you have kind of that your project has, or the findings that your project has as kind of recommended. But what kind of difficulties do you see on the ground for nurseries, for anybody involved in the industry in terms of kind of fighting against the kind of prevalence of Phytophthora in water and so forth?
I mean, it is hugely challenging, and it's particularly challenging for those in sort of southern latitude countries where water is a scarce resource, and they tend to recycle. But there are a range of different water treatment options available, and I suppose you essentially, I think that it would be easier for larger turnover nurseries to implement the necessary changes, because obviously there's a cost associated with it. But what I should mention is one of our Scottish nurseries, as part of this project, when we reported back to him the amount of Phytophthora that we were not just finding the DNA of, but also baiting into live culture from a reservoir from which he was actually watering his plants. He actually went to the expense of sinking a borehole to extract his water from sort of clean borehole water. So, I mean, there was a clear evidence of a management change. So I think if you can demonstrate effectively to nursery managers that this is a particular problematic area and they can clean up their Phytophthora load by dealing with water, either new water source or putting in water treatment measures, then they'll be much less liable to losses due to Phytophthora. But I also think that one of the biggest issues we found was a lack of awareness. Lack of awareness of symptoms, and a lack of awareness of the fact that disease symptoms were caused by Phytophthora, not by other factors like the plants dried out or they got frosted, these kinds of things.
So it's a kind of an education component to it as well. It's just kind of
Yeah.
educating the people who are working in nurseries about the dangers and how to look out for the signs of Phytophthora.
Yes,
Mm.
and we actually we had held two one-day seminar sort of nursery training days in one in England and one in Scotland, not just as a result of this project, but a previous project, Phytothreats, where we developed the methods. And we had quite a good uptake of that, where we really ran through the water treatment issues, how to deal with your growing media waste and your green plant waste. And above all, what does a diseased plant look like, and what can you do to deal, how do you deal with it? How do you diagnose it? Then how do you effectively dispose of it? So
Mhm.
yeah, I think it is around training and awareness raising.
Sure. And so just a couple of questions from our audience. So somebody is asking, did you find any Phytophthora species on eucalyptus plants?
Yeah. So interestingly, we did sample eucalyptus from in the UK, but that was the only eucalyptus that we were that was sampled, and it was from a garden centre associated with the nursery. And I think we did find Phytophthora, but it wasn't obviously symptomatic, and it may have been in the potting root or the mix. And I can't remember which species it was. I'd have to go back and look at our output. But yes, we did, but I don't think it was anything particularly damaging, because obviously you've got some Phytophthora which tend to exist in water. You don't want any you want to reduce all your Phytophthora in nurseries, but there are some which are more damaging than others. Just to, in quick.
Excellent.
But yeah,
Uh,
short answer is yes.
and another person is asking, Dr. Green, did you note any recovery of Phytophthora species via baiting or metabarcoding?
Did I note any
Did you note
recovery?
any recovery of Phytophthora species via baiting or metabarcoding?
When do you buy Phytopithium? Sorry. Yes. Yeah, we got a lot. So this was, did we notice any fight? Is this correct?
Apologies,
It's Phytopithium.
Phytophthisium. Yes, apologies.
Yeah, that they were quite common, and we have found a lot of phytopithiums in other projects in growing media as well, and isolated. But yeah, I can't off the top of my head remember which species, but I'm pretty sure we you do tend to get quite a few pythiums when you do baiting, and some of these are phytopithiums, and you get Phytophthora as well from baiting water. But yeah, in short, yeah, we found there were phytopithiums, and they seem to be quite prevalent.
Mhm. Excellent. And one last question. What treatments do you suggest for open water or recycled irrigation sources?
Well, it really depends on the setup, and I'm no expert on that, but I know that there's like slow sand filtration, there's UV, and there's different chemical treatment. So I think it would be a case of looking, if any nursery manager looking to see what the expertise and what other nurseries are doing in their region, and going from there, because obviously it requires an infrastructure set up and that sort of thing. But as I said, it's physical filtration and then chemical treatments as well. But there are a range of different options depending on your setup.
Excellent. And sorry, just one last one comment from our audience. It reads, thanks a lot for the excellent presentation. I'm wondering if the project also includes the search of pathways of introduction of Phytophthora in the different countries. Example, trade of seedlings between nurseries. Thanks.
Yeah, I mean that was one of the original aims of the project, was that we were to test plants that had recently been imported, but no, essentially it became too complicated when you're working with quite a large number of countries. So we wanted to keep it simple, and we just said to each country, could you please go to a nursery where at least one of them is importing plants from overseas? And we will be testing those plants, but it wasn't testing specific pathways. I think that would be the next step, to be honest.
Sure. Sarah, thank you very much.
Right.
We could fire another 10 or 15 questions at you. Loads of questions coming in the chat chat and Q&A box there. But unfortunately, we just have to move on to our next speaker now. Thanks very much once again, Dr. Green, for joining us today, and thank you for such an excellent presentation. Okay, so next up we have Marleen Botterman joining us. Now, Marleen is currently a plant virologist at the Dutch National Plant Protection Organization, where she specializes in plant virology and molecular biology. In particular, Marleen's research focuses on plant pathogen diagnostics, playing a key role in the detection, characterization, and management of plant diseases caused by viruses and viroids. She has contributed extensively to the field with key publications on topsoviruses, viroids, and other pathogens. Her widely cited publications appear in journals such as Archives of Virology, Plant Disease, Phytopathology, and PLOS1, showcasing the impact of her research in both applied and fundamental plant virology. Today, Marleen's talk is entitled Baseline Study of Virus Reservoirs in Native Plant Species and Crops, which is also part of the UFresco Phytosanitary Research Initiative. Marleen, thank you very much for accepting our invitation for a webinar series. We're delighted to have you with us today. So the floor is yours. I'm going to stop sharing my screen so you can start sharing yours. Thanks very much.
Thank you, Barry. And thanks also, Sarah, for this really nice talk. It's really excellent. I'm going to start share my screen. Please shout out if it works.
Yes, that looks great,
so
Marleen. Thank you.
You can see my presentation. You can hear me. So thanks for the introduction. I'm just going to move your heads around a bit. Otherwise, I can't see my slide properly. So my name is Marleen Botermans, as Barry already told you, and I work as a plant virologist at the National Plant Protection Organization of the Netherlands. And in a couple of 50 minutes, next 15 minutes or so, I'll be talking about virus reservoirs of both wild plants, trees, and crops, and why I think it is important to have insight in these virus reservoirs. First, I'll give you a short introduction of the organization I work for, the National Plant Protection Organization, and why for us it's also very important to have insights in these reservoirs. And I'm talking about viruses because I'm a plant virologist, but I think the same story might also somehow apply for species like Phytophthora or any other organisms. So this is the, can you see my pointer as well, if I move it around? Yeah, excellent. So this is the organization and the buildings I've been working at for 17 years now. Here, our office building in Wageningen, our quarantine laboratory, and our quarantine greenhouse facilities, where we do our some research, and we have our experiments, and we have a really large living plant collection, also with many quarantined species. The NPPO is a governmental agency, and it monitors plant health, in this case in the Netherlands. And we provide guidance on phytosanitary policies and actions, and all these are based, in our case at least, on regulations of the European Union. Now, to be a bit more specific, we have a responsibility as an NPPO that the plant materials both entering and leaving the UK, for example, to leaving the EU, for example, to the UK, are free from harmful organisms. Our aim is not only to safeguard agriculture, but also biodiversity and international trade. And we do so by monitoring and surveillance, for instance, to detect potential or real quarantine organisms, and via plant pest control. And we contain outbreaks of any quarantine organisms or other harmful pests to prevent spread. Now, I just mentioned quarantine organisms. For some of you who are not aware what this means, these are harmful organisms which are included in official quarantine pest lists. And these lists are again part of the EU plant health regulation in our case. But also other territories like the United States will have their own official quarantine list. It's like a blacklist of harmful organisms. And the species on the lists will differ among each other. There will be some overlap, there will be some differences too. So we're dedicated mostly to the species on our EU list. But what are the criteria for listing from these harmful organisms in a quarantine organism? To be placed on such a list, the identity should be clear. Unfortunately, that isn't the case for some of the species that are now listed on the EU quarantine list. But this is actually a criterion. It also, this organism we're talking about should be absent, in this case in the European Union, in the territory, or it can have a limited distribution, very limited. Another criterion is that it can establish and have economic impact, and that measures are to prevent introduction are still feasible. And I was at the previous slide also talking about potential quarantine organisms. These are harmful organisms that may pose a future risk of plant health, but are not on a list yet officially. And these may be under consideration for inclusion on such a list. But then first, the best risk analysis needs to be performed. Yeah. So this is some background information of what an MPPO does and why. And there's much more to it, but I just highlighted some parts which I think are relevant for the rest of this presentation. Yeah, in case you were not aware. So the Netherlands Institute for Factors Invasive Plants and Plant Health, that is the department I work for, and it is part of the MPPO. And we provide expertise on 7 different organism groups, including the plant viruses. And we are national reference laboratory for the Netherlands for all these organism groups. And for both bacteria and plant viruses, we are also part of the European reference laboratory. We provide information on taxonomy, biology, epidemiology, name it, to the other parts of the MPPO to support plant health. And we perform plant health diagnostics. And we do this diagnostics on samples, on various sample types, the ones we receive from official surveys, plant health inspectors that go into the country and do screenings and take samples. We analyze these, but also from import inspections. Think of inspectors at the border or in the harbor, and checking out solanaceous fruits, for instance. We receive material from companies and growers that may be diseased, and this is like plant clinic samples. And lastly, we have quite some samples from research projects, like the virus reservoir studies I'll be talking about shortly. Yeah. At our institute, we have have been using various test methods to detect and identify viruses. Of course, ELISA, bioassay, various PCRs. But since 2016, we've really moved to using high-throughput sequencing for the majority of our testing. And I'm sure that most of you will know why high-throughput sequencing is so nice of a technique to use in plant health and what it does. But let me shortly explain for those who are not aware. It's really a nice technique because you don't need any prior knowledge of the viruses that may be present in your sample. That's because everything is sequenced with this technique. So all sequences of both known, but also unknown or putative unknown viral species, viroid species. Yeah? So it's very useful for us. And since a few years, we have a HTS test for detection and identification of viruses and viroids under ISO 17025 accreditation in our lab. We're very happy with that. What we're not so happy about, maybe, is that due to using this new test method, we've been having many more virus findings than before. It's different than using more targeted tests like ELISA or specific PCRs, because we find a lot of unexpected stuff out there. Quarantine species, I was just mentioning about, but also non-regulated and putative novel species as well. I keep moving around, sorry, because there's all these faces in my in my view. Sorry. Can I make this smaller? Whatever. Also important to know is that these findings of these viruses are in plants that not always have typical virus-like symptoms. So we realize ourselves much better than before, I think, that also healthy-looking plants may harbor various viral species. And before, I think not much effort was put in those type of plants and finding out what's in there. These are, in this list are the virus species that have a quarantine status in the EU and that we had outbreaks of in the past 2 years. And in green, those are the findings we had, or the detections we did with HTS, high throughput sequencing. And actually, all of these were quite unexpected findings for us, also from virus reservoir samples, and we were quite surprised that we'd find these. We also find in many cases, for example, hibiscus with cowpima model virus was a novel, a new host also for this species. There were 6 other viruses present and 1 viroid in this particular hibiscus plant. And also a lot of new species, known species, known, but not known previously from hibiscus. I've been saying the word new already quite a bit. Due to HTS, we have many new virus findings. And with new, I mean new for science, so putative novel species, a new host, like the hibiscus I just mentioned, or new for a territory. Maybe the first finding for the Netherlands. But interesting is to know, is this also really new trouble? Are these potential quarantine species and are phytosanitary measures required? Or is this simply lack of knowledge of the virus reservoir? And are these species already widespread? Are they neutral, even maybe benign? And are phytosanitary measures not required? So this is where finally the virus reservoirs come into the game. We think they are very important to get this knowledge, to get this baseline information, which is essential to assess risks, but also to guide actions if necessary. And it provides information we need to know what viruses are present in your territory, both known ones, because you're often not aware of the presence of these, but also putative new ones. What are the host plants associated with the viral species? What's the geographical distribution? Is there a relation with symptoms or not? And virus reservoir studies are nice, but I think it only works well if it is done together with international collaboration. And I think impact can be maximized through data sharing. We call it pre-publication data sharing, but it's like reaching out to your partners in various ways. Like, hey, I found this in this crop or in this wild plant. Have you found it? And then exchanging information and finding out that maybe everybody finds the same virus species, but maybe in different host plants. Sometimes not associated with a disease, sometimes they are. And I think with this, we get insight into the geographic distribution, epidemiology of particular viruses. And it may present even unnecessary regulation. And also very important, of course, it helps to anticipate outbreaks in new areas. And this is where I introduce a really nice Ufresco project I've been involved in, which I coordinated it with Adrian Frox from Para. It's called Virus Reservoirs. And in this project, 9 members work together and everybody generate and share data from both surveys and diagnostics and other virus reservoir studies. I want to give you one example of a really nice virus reservoir study. and it's about a Podex virus. It's called plantain virus X. And this species was described quite some time ago in the United Kingdom in Plantago lanceolata. It's a plantain. And it was described by John Hammond. Maybe some of you will know him. But then a few years ago, he got this note from the ICTV, the International Committee of Taxonomy of Viruses, and he said, hey, well, there's no supporting sequence for your species, which was otherwise well characterized, but at that time it was just simply not sequenced. And the species risks removal of the list because there was no sequence. So John Hammond reached out to several people, and we've been all looking for the plantain virus X sequence. And we've been sequencing some plantain plants. But let me first introduce to you another Podex virus, Actinidia virus X. This species was first described in kiwi fruit in 2011 in New Zealand. And a few years later, in 2016, the same virus was detected in blackcurrant plants in Canada. And as it wasn't known from the EU yet, and it might be a novel species in Ribes, it was recommended in the EU for quarantine status. But then, yeah, I just told you we've been sequencing some Plantago lanceolata plants, and there was an independent study by Wageningen University also sequencing some Plantago plants. And independently, we identified a Podex virus in these. And after sequencing, it appeared to have the highest sequence identity with this Actinidia virus X. And then a really nice piece of the puzzle came because at Ferra, they discovered the samples from Plantago from the 70s, which were studied by John Hammond. They found it in the freezer, and they were able to sequence these historical isolates from the 70s. And guess what? There was this Podex virus in there, of course, but it also had highest identity with Actinidia virus X, which is logical because there was no sequence from plantain virus X available yet. So we were able actually together to solve this taxonomic issue. Actinidia virus X is now officially a synonym of plantain virus X. The name is saved, and it's officially recognized by the ICTV now. And during this nice collaboration with various partners, various countries, more people came involved. There were additional findings in different crops, different wild plants from different countries, including European countries. And at that point, the criteria for listing as a quarantine organism were not fulfilled anymore, like the criteria I talked to you about before. So we collectively informed our policymakers, and we were able to prevent, in this case, an unnecessary regulation. Yeah. And this, if you want to know more about this, is a nice paper, Plant Pathology, which was published. Now, and no time's running out, but just a few examples of virus reservoir studies we've been doing the past few years. There's one in imported selenaceous fruits. We want to see what's coming into the country, and instead of ELISA PCRs, we've been sampling and sequencing 300 samples of various types of fruits. We did over 200 virus findings, belonging to over 20 plant virus genera. And just a small example of one pepper imported from India. We did not only find the cucumber mosaic virus, but various polariviruses, spottyvirus, and even a begomovirus in this one. So often it's quite a zoo in these samples. And there's a whole other story if to assess what the risk of all these viruses are for agriculture in the European Union. It all depends on the contextual information also. Then there's another nice project we did under the Virus Reservoir theoretical project, where I've been sampling and sequencing 1,500 plants from various wild plant families. And in these, which were actually without typical symptoms, so plants look really nice and healthy in the field, in the country, we've identified over 300 different viruses belonging to over 53 plant virus genera. And one example is the dandelion here, in which we found various species known from that, a close relative. Also a virus which has been causing some problems in lettuce over the past years, but we never certain where it came from. But it appears that dandelion is a host, and we also identified new species in this one. Another nice Ufresco project, Virno tree, which started previous year, is on trees and what's in those, what viruses are present in trees. We've been sequencing now 100 tree samples, and I just got back to the results, and we have some 15 virus findings, so much less compared to the wild plants until now, but still really interesting findings. Here an example of an ash, in which we found 2 known species for ash, but novel species for our territory, so it's nice first findings. But also new species of the family of Segroviridae, which are also niboviruses. And I can go on for a long time, but these are some of the nice publications which were published in recent years and are involved in virus reservoir studies. Some also that resulted from the virus reservoir Ufresco project. Some have angles like what's out in our territory. Some are focusing on wild plant families or crops like carrots, and I expect there will be a steep increase in many of these. Nice project with a lot of collaboration and data sharing, and I think this will help the field a lot. And now I've reached the final slide with some conclusions. Well, it will be clear now that for appropriate phytosanitary action, a lot of information is needed. It's important that nations gain insight in their virus reservoirs, both in crops and wild plants, and both in plants with symptoms, but also asymptomatic plants. HTS is a good method to gain insight into these virus reservoirs, because you can so broadly active sharing of information is important, and it helps to combine existing pieces of information for broader insights, and it may prevent unnecessary regulations that no one wants. I would like to thank you for attention. I would like to acknowledge the virus reservoir partners from Ufresco, our virology and molecular biology group, Adrian Fox, Sebastian Massar, and John Hammond. Thank you.
Thank you very much, Marlene, for such an interesting and insightful presentation. I just want to remind everyone that the chat box and Q&A section is open, so please do feel free to type a question for Marlene at any point between now and the end of the webinar. Marlene, just while we're waiting for a couple of questions to come in, I was just fascinated by the kind of breadth of the samples that you're incorporating into the project. And I was just wondering, I was very curious about species selection. So my question is, how were the specific plant families and species chosen for sampling for the project? Or what was the kind of criteria you used for those particular samples that you used for the project?
Well, the data I presented were from various projects. For the Ufresco virus reservoir project, I know that the members had their own selection criteria. Some were, for example, focusing on hops, and they just been sequencing hops all over the place. Others were more interested in what's out there in solanaceous plants and crops in our country. For example, I gave the example of the wild plant species, and for these, we selected species that are quite general, growing near agricultural fields mostly, so common wild plant species, which are quite abundant, which can be found in various places. So we could also compare these, find different species near beet fields or near potato fields. So that was one criteria. And for the solanaceous fruits, we were basically looking at all fruits, solanaceous type of species, fruits that are coming into the EU. That's so basically all. You're frozen a bit, or am I frozen? No, it's okay now. And for the trees, actually, we looked at trees in which phytoplasma or viruses were already identified before in other countries. And often, like, we had no information on these, but I expect those viruses to be present in our territory as well. But if you don't look, you don't find anything, right? So I also want to establish that the stuff the Germans find or the English find, we have probably too, because we're so close. Yeah.
Excellent,
Yeah.
Marlene. Thank you so much. Just a couple of questions from our audience. So somebody is asking, do you deal with any mushroom virus samples in your clinic? And if so, is HTS the preferred method of detection in this case?
Oh, mushroom viruses. I think 17 years ago we did a bit of these, but it's so complicated. No, I think the identity, if I'm not mistaken, and I'm not an expert because I don't do anything with mushrooms, but I think identity is quite difficult. So I think HTS can be applied to mushroom viruses, but first you need to know what you're looking at and what species are out there to get an idea. And I don't know. I don't, this stage, know anybody dealing with mushroom viruses. I'm sorry.
That's okay, no problem. Marine, another person is asking, based on your results, do you think that HTS methods are accurate for identifying viruses at the lowest taxonomic level, or do you combine them with other techniques?
No, actually, I think they're depending, like everybody, there's different HTS tests, and you need to validate them for the purpose and the scope you're using them. But HTS by itself is well sufficient if you take the proper checks and controls and you know what you're looking at as a sole method. So we actually don't even not in critical cases, we don't do any other methods if we have HTS, but we check a lot of contextual information to see if it's right. But it's a really nice technique for identifying species at the lowest taxonomic level, even below genotypes. Yeah. Is that a clear answer? Yeah.
That's perfect. Marlene, again, just in terms of future directions, probably this will be the last question now, we're just pushing up on 10. In terms of future directions, what are the anticipated next steps following the completion of the project, particularly in terms of research and its implication for policy?
Well, one big implication for us as researchers is at this stage, time and priority, because the sheer amounts of viruses we've identified, and then I only mentioned to you like the relevant virus findings. There's so much time gets involved in properly annotating all these sequences, all archiving, analyzing all these findings, and then publishing these. I think there's like decades of years work for it. And I'm a bit worried about it's nice to have this technique and it's really important to do these virus reservoir studies, but then there's not enough plant virologists and not enough time to get all this data out there, if you know what I mean, and properly inform our policymakers and our risk assessors. And so now I think they get just bits of the puzzle, which we think are critical cases and should have priority, but there's just the tip of the iceberg. So I think it would be really nice to have maybe AI or other ways involved to speed this process up before we can really, I think it will be a next step.
Mm-hmm. That's super. And just one final question's just come in. It just reads, thank you for the nice presentation. Are you identifying dsDNA virus genomes via HTS DNA sequencing data analysis?
Also, we do RNA-seq, but then also from DNA viruses, they have this RNA stage, and then We often don't get the complete genome sequence, but if we find some context, some sequences of DNA viruses, we do DNA-seq as well. And yes, we can identify these as well.
Sure. Thanks very much. Marlene, I think we'll just leave it at that now. It's just past 10 o'clock. Yeah, I'd just like to say thank you very much once again to Dr. Sarah Green and Marlene Botterman for giving us wonderful and insightful presentations and providing us, I think, with such great overviews of their respective projects this morning. Our next webinar will be on January the 23rd, 10 a.m. Central European Time, when we'll continue our new webinar series on plant health and protection. I'll be sending the invitation out to all of you in the next couple of days. In that breakfast club, we will have Dr. Hugh Evans from Forest Research in the UK talking about range expansion of bark beetles in the genus Ips, and Dr. Pareem Portier from INRAE talking about building a reference database for plant pathogenic bacteria identification by MALDI-TOF and evaluation of the method. Again, thank you very much to our panelists for joining us this morning, and thank you everyone for attending and for your questions. We really hope you enjoyed the webinar, and of course, we'll see you again next time. So thank you very much, everybody, and have a good day.
Thank you. Thank you.
Thank you. Bye-bye.